So, welcome everyone to the Sea Grant Water Resources Vision Team's webinar series. As you're joining us, we want to give you a chance to get used to the polling technology that we'll be using throughout the webinar, which is Poll Everywhere. And so we just have a simple question for everyone. What interested you in this webinar? It's really easy to respond. You can open a browser on your computer or on your phone or other device and go to pollev.com/SOTS549, which is said there at the top. And then you can just type in your answer. And so we thought that this would be an interesting way to start off the webinar and give people a chance to sign in. We know it takes a little while on Zoom to get everything connected. Welcome everyone. Nice to see the names coming in as everyone is joining the webinar. So I'm Stephanie Otts. I'm the director of the National Sea Grant Law Center. Just to get participants familiar with the polling technology that we'll be using for the webinar, we wanted to start with this initial question about what interested you in the webinar. It's easy to respond. You just need to navigate on your browser, on your computer, phone, or other device to the website at the top of the screen, pollev.com/SOTS549, and you'll have the opportunity to submit your answer to this question. Welcome everyone to the Sea Grant Water Resources Vision Team webinar series, COVID-19 in the water environment. As people are joining the webinar, we are providing this opportunity to get used to the polling technology that we'll be using throughout the webinar. And so if you want to navigate on your browser to the website at the top of the screen, that will be how you will be able to respond to poll questions throughout the webinar. And it's already so interesting to see what has brought individuals to the webinar today. So we're going to get started here in just a couple minutes, but wanted to give folks a little more time to get logged in and connected to the audio. So thank you for joining us. Okay, well, I'm going to close the poll question as we see answers still coming in. And if you're entering them, they won't be lost when I go to the next question. But this is great to see all of the interest. So, as I mentioned, my name is Stephanie Showalter Otts. I'm the director of the National Sea Grant Law Center. We're based at the University of Mississippi School of Law, and currently part of a leadership team that is helping to coordinate and facilitate individuals within Sea Grant programs working to address water-related challenges. And we call ourselves the Water Resources Vision Team. We have been working for a couple of years to do strategic visioning around water issues in Sea Grant. And some of our key themes that have emerged are around environmental and human health, water infrastructure, land management, restoration, and development, and water planning and socioeconomics. And so we have about 50 people within the Sea Grant Network as part of the team right now, but we welcome participation from anyone in the Sea Grant Network. So if you are interested in learning more about the Water Resources Vision team or joining us in our activities, please send me an email at the email address noted on the screen. And the image on the screen was just a word cloud from one of our first team meetings where we asked people what they were working on. And so you can see the range of activities that are covered by our group. So this is the first webinar in our 2021 webinar series, which is new for us to do some webinars. We have a couple of objectives, which we hope to accomplish today and throughout the rest of the year. One is to raise awareness and visibility of Sea Grant water resources programming within the wider Sea Grant network. We want to highlight innovative Sea Grant research, extension, education, and communication activities related to water resources. And then we want to provide opportunities for networking and strategic thinking around pressing water issues. And so we hope that through the presentations today, the use of the polling technology and the time that we've dedicated to have some kind of thought-provoking questions as part of the Q&A at the end of the webinar, that we'll be able to start accomplishing some of these objectives. Just for an overview, we have 4 presentations for you today. There will be an opportunity to ask clarifying questions after each presenter, but we want to hold kind of bigger picture questions until the end when we do have time dedicated for a Q&A. And before we get started, I really wanted to give a big shout out to the planning committee for this particular webinar. And Karen Bareford is the National Water Extension Liaison at the National Water Center. And we've been providing support to logistics support and other planning support to help the webinars happen. And then Sam Chan, Kelly Donaldson, and Jennifer Hawkswell were kind of the brain power behind the structure for this particular webinar. So it took a lot of hands to make this happen. And so with that, I want to turn it over to Sam to give us a little bit of background about kind of the vision for this webinar and conversation.
Thank you very much, Stephanie, and thank the Water Resources team and the Sea Grant program for enabling this webinar series. So back in March, I think most of us were facing the early stages of this pandemic. Lots of uncertainty. Most of us were going through major changes in our lives and our programming. Many of us were working from home. And there are many questions that we in Sea Grant, as an extension agent, section specialist, researcher, we're asking ourselves. And number one is the safety of our colleagues, the safety of our family. what's the impacts of COVID-19 directly on us and the working environment that we had? So we had many questions, but yet there was just so many unknowns that we really couldn't come up with a good research outreach program that was going to be accurate and consistent, and really one that acknowledges the uncertainty involving science. And so with that, we started putting together a series of questions and needs that we thought would be important. And we started working with Dr. Chris Stepanek in really working about what about people who actually work in that water environment, like water quality volunteers, how might they be impacted? Does COVID-19 even exist in the marine environment, our estuaries and our beaches? And is there any exposure risk? What about the shellfish and the fish that we work with? Many questions. And then we read the article that Dr. Prather had to address, is the issue of aerosols in the environment. And so with that, we had many questions. There are a lot more questions than we had answers. And I'm happy that we now have a webinar that begins to address many of those questions so that we can use that to drive outreach and prime really what Sea Grant can do in terms of our research as well. So thank you.
Thank you, Sam. And Sam, did you say the cartoon was drawn by one of your students?
Yes, that was drawn by Aaron Cathcart. During the early stages, we just didn't know much about COVID-19 or in the water. And so he drew this cartoon to get us to think, and that got me thinking a lot about COVID-19.
Thanks. Right, so our first speaker is Dr. Kimberly Prather at the University of California, San Diego, and we're really excited to hear more about her research and have her kind of set the kind of a broader stage for some of our conversations later today. So with that, Dr. Prather, I'll turn it over to you.
Well, thank you. Thank you for having me here. Yeah, and I want that drawing for my talk, as you will see. You know, it has been, I have to say, a bit of a bumpy ride in sort of getting people to think about what happens. You know, I noticed in the word, you know, all the words, there was nothing about air, and I study the air. And what I will, you know, I think hopefully make clear today is that once things get launched into the air, which they can from water pretty easily, a lot more people are exposed. So I would like to expand the thinking a little bit beyond, inclusive of, but beyond, you know, what is in the water. And so one of the things what I'm going to talk about today is just what is going to happen, what is happening, you know, when we get these massive, you know, atmospheric rivers that are leading to runoff and storms, and how is that influencing sort of the environment and in particular the air that we breathe. So I am the director of a National Science Foundation Center called CASE, as we affectionately refer to it as, the NSF Center for Aerosol Impacts on Chemistry of the Environment. And so again, just interest of full disclosure, my research is on the air. I know a lot of people are here to hear about the water, but they're connected. And I hope everybody will be in agreement on why it's important to pay attention to both. So next slide. All right, so this brings me to what is happening here in San Diego, where I live. This is a picture taken by Mark Merrifield on the upper left quadrant of what is happening when we get hit with these massive storms, which are becoming very much more frequent, where we get you know, a lot of flooding and a lot of runoff into the ocean. And so what I will talk a little bit about, something that we've worked on with oceanographers, I don't know why I got cut off, I'm sorry, a project on the upper right quadrant that has to do with Seaside, which is looking at, we sort of mimicked what happens when the stuff pumps into the ocean, what can get in the air. And so, you know, a big focus of my research is trying to understand, of all the pollution that gets pumped into the ocean, how much of that gets into the atmosphere? And this can be in the form of viruses, bacteria, or toxins for that matter. Next slide, please. one thing we see when we go to the beach is time to shut down the beach, right? You can't go swimming, you can't go surfing, but no one tells you can't go breathing. And I will argue, and you'll be, I think, more cognizant of the fact that this stuff, when it's that enriched in the ocean, can get transferred to the air, largely in regions where waves are breaking. Waves are just great at launching, viruses and bacteria into the atmosphere. This is not a surprise. This has been shown since like the 50s that things that are biological, so viruses and bacteria, really don't want to be in the water. They're what we call hydrophobic. They hate water. So they float on the surface of the ocean, and when waves crash, this stuff gets launched into the atmosphere. And that is a major focus of my research program. Okay, next slide. So this is what it looks like when sewage here where I live by Imperial Beach flows into the ocean. This is coming from the Tijuana estuary. This is a time when 143 million gallons of sewage just dumped into the ocean. These episodes where sewage, raw, untreated sewage gets into the ocean happens a lot more frequently when you have these huge rain events as well. More discharge, more water, hitting the surface of land and sort of building it up will lead to these massive events where you get lots of sewage into the ocean. Next slide, please. So I mentioned a major study that we got involved that we piggybacked with Sarah Giddings and Falk Feddersen on a project where they put dye into that pulse of, there's a river and an estuary that flood into the ocean. And this happens all over the world. It's not just San Diego, but they put dye into this estuary and looked at where it went. And the focus of the oceanographer study was to look at how far up and down the coastline this pollution went. You can see the pink dye in the ocean. And so basically, our question was, click it one more time. If you have this unique dye, can you detect it in other places? In other words, inland locations, not just if you're breathing it on the beach, but if you're at other places in San Diego County. Next slide. So as I mentioned, I'm the director of a large National Science Foundation center. So this is related. This is the facility we use at UC San Diego. It's called the H Lab or hydraulics lab. And so basically in this facility, we can pump seawater in and we can break waves and look at the viruses and bacteria that become airborne. And we've been doing this for now 10 years. And I won't go into the details of the results on the left, but we published a paper that's showing that certain bacteria and certain viruses become heavily enriched in the atmosphere, which means they can blow over like large regions of San Diego and potentially be inhaled. Okay, next slide, please. So, having said all that, with a center, with a focus and interest in, what is in the surf zone, what is getting flooded into the ocean. When SARS-CoV-2, when this pandemic hit, I was contacted by the National Science Foundation and asked, can you help? We know that SARS-CoV-2 can get into wastewater. We know the wastewater can get in the ocean. We know that, sewage, pollutants in the ocean can become airborne. So the question we were asked by NSF is, can you study it? Can you look in the air and see if you're where are you detecting SARS-CoV-2? Are you seeing it? And there's you've seen the map, there's a beautiful estuary and a river. It goes in the ocean. Do we detect it along that path? And then do we detect it in the air? And so this is a wide open question that nobody had thought about. It's kind of odd that nobody had thought about it, to be honest with you, because SARS-1, which was in 2003, was a sewage release, a leaky sewer pipe that got airborne outdoors and got into buildings and made people really, really sick. So we know that this virus, SARS-1, SARS-2, they're both in sewage, wastewater, And the question is, can they become aerosolized and make it into the air? And so one of the questions we had was, this is a direct feed from Tijuana, where there's a lot of cases, unfortunately, of this virus being in, people are infected. And so, one of the ways that people monitor the relative, impact of this virus is to look at wastewater. And we're doing this at UC San Diego, which I'm happy to talk about, but you can use this as a major gauge of community exposure. And one of the things we know is that in Tijuana, the exposure is really high. Next slide, please. So we put sampling sites, things moved. Sorry about that. We put sampling sites for air and water all along the coast to try and look at, what's in the water, what becomes airborne, and how, what is getting dumped into the ocean might make it into the air. Next slide, please. So this just shows where the Tijuana, river flows into the ocean. And so we're looking for conditions where we know the flow is high, which quite honestly, most of the study that I will show you was done during a time when there wasn't a lot of rain. So turns out people have shown for other pathogens that you get more flow if you're in a time period where you have a lot more rain versus no rain. So there's a dry versus wet season difference. When it's wet, which is where we are right now, there is a massive amount of flow into the ocean, and that is when we expect to see things. But we decided before that happened, let's just look before we get to the conditions where rain is playing a major role. Next slide, please. So this again shows the sampling sites. We've had many citizen, scientists volunteer. One of the things we were planning to do before the pandemic hit was we were going to swab surfers when they went out in the ocean. We didn't get there. We would still like to go back to doing that. You can say stuff is in the air, but what are people actually breathing into their bodies from the environment? That is a wide open question, again, from everywhere, not just San Diego, anywhere coastal. This is a serious issue. So this just shows, the way we collected the air, the way we collected the viruses into what is called a biospot sampler. It's a special sampler that collects things in a very careful way to not just destroy the virus. But there's a multi-pronged approach to look at the water and the air and to see where this virus is going and see where we can detect it in the environment. Next slide, please. So hot off the press. We just submitted this paper, and the answer is, you can see at the top, detected, not detected, looking at the river water, estuary water of the ocean and sea spray. The good news is, so far, we have 90 samples in the air and we've not detected it. That is really good news. The bad news is that we do see it, a little bit in the ocean, only one time because it's pretty diluted. We expect that to change with wet season. But we do see it in the estuary and river in high quantities. I was shocked. My students were only pulling in like a little bit of sample, and the quantities in the estuary and the river were quite high and easily detectable. So it's out there. The question is it out there enough to make it in the air where people can breathe it? So now we are, we are sampling the air more as we move forward, and I'm happy to answer questions about that. All right, next slide, please. So I think at this point, I'd like to just thank everybody. This is a different thing, different slant than we normally think about from a Sea Grant. But if we care about human health and impacts of pollution, the air, just once it's in the air, and this is what we're seeing for this virus, once we share, this virus is airborne. And once we share the air, we're all affected, and many more people can be exposed. And so, I think, the ocean and atmosphere are connected, and I would like to see more research along these lines to address these issues. We will be ahead of the curve, and I think lead research worldwide. This is a worldwide global problem, and there's a huge team of us working on this that has made this research possible. I'd like to thank everybody that's been involved so far, and I'd like to thank you for inviting me to give this presentation.
Thank you so much. That was so interesting. And so Karen has been monitoring the Q&A. I think we have a minute or two if there are. Karen, any clarifying questions?
We do have a couple of coming in. The first was, thank you for all of your work, Dr. Prather, and were the SARS-CoV-2 viruses detected in the ocean and estuary confirmed to be viable, or was it just RNA that was present?
Just RNA. We have not gone to the viability. Right now, we're killing it on purpose. That's the next step. Once we find hotspots, if we do, then we have the people that do infectious disease at UCSD in the right facilities. They're lining up to do the analysis. But right now, we're just doing a broad survey of where do we find it, if we find it.
Thank you very much. The next question is, can this virus survive in oceanic salinity?
You know, nobody has studied that directly that I'm aware of yet. Let's just say we hope not. You know, can it, I would broaden that question. Can it survive directly in ocean salinity? Can it derive in sunlight? People are looking at this. It doesn't really like sunlight. So we hope like heck that, you know, when it's out in the environment that it's just killed right away, either through, as you say, the ocean salinity or sunlight. But those studies are yet to be done. That I have not seen anything published on this.
Okay, thank you. The next is somewhat related to that. Is the virus that's in the marine environment possible for viable transmission?
So it's back to the question of, is it viable? Is that correct?
Yes. Do they know if it's viable for actually transmitting?
No, I mean, it has to be viable, it has to be alive. So all we're looking, I just want to be really clear, and I thank you for that question. All we're looking for is the presence of the virus, where it is. And I can tell you it's high concentrations, whether it's alive or not, or viable or not, infectious or not, we don't know. That is the next step. And that is what we're working on now. We are working, as I say, with infectious disease doctors at UC San Diego who are looking, you know, at this problem. It's really hard. I mean, it turns out that to go into what's called a BSL, it's a basic, like, you know, it's a special facility, very expensive facility to test it, to do live virus and to look for it. And so we are waiting to make sure that we have enough, that we're seeing it enough before we move to that, you know, sort of level of looking for the virus. If we start to find it everywhere in high concentrations, certainly we're going to be looking to see if it's infectious, but that is the next step.
Okay, thank you very much. Stephanie, we have several more questions coming in. Do we want to try and answer these, or should we move on for right now?
Yeah, I think we should move on for right now. We will be capturing all of your questions. If there's time at the end, we might be able to circle back, but we can also follow up after the webinar. So yeah, thank you for your questions, and we'll move to the next presenter. So our next presentation is Dr. Richard Melvin at the University of Minnesota, and we're kind of moving from the ocean side to freshwater. So I'll turn it over to you.
Okay, thanks, Stephanie, and thanks for inviting me to talk today. My name is Rich Melvin. I'm at the University of Minnesota in the medical school there. I'm actually more of a geneticist, and this is not my usual subject matter, but when COVID struck, one of my colleagues and I were thinking about how to serve communities that were kind of underserved in medical services for testing, et cetera. And so we began a program of wastewater monitoring for COVID-19 in Minnesota, which now covers the entire state. And Minnesota Sea Grant found out about that, and they had been fielding questions from people in Duluth, Minnesota, which is where I'm at, on Lake Superior, from the community that deals with a lot of tourism, with their concerns about safety for tourists. And they asked me if I could collect water at local beaches, which is one of the great draws of Duluth, Minnesota in the summertime, and test that for the presence of the SARS-CoV-2 virus. And so, sure, why not get out to the beach rather than in the lab? And so we started sampling. So what you're seeing here in this opening slide is on one of the local beaches in Duluth, kind of a neighborhood beach, Rocky Beach, on 4th of July. And 4th of July is beach day in Duluth, Minnesota. Tons of people out there, but we're in the middle of a pandemic. And as you can see, I don't see any masks present on these people. So there's a lot of concern about whether we can monitor what was happening in the beach population via the water. So next slide, Stephanie, thanks. So, it's the question of what's going on in the population, and is it reasonable to go monitor at beaches? And of course, a lot of us know that beaches are monitored frequently. There's a program in Minnesota that monitors Lake Superior beaches for fecal bacterial contamination. And when they're monitored, about 12% of the beaches on the Minnesota shore of Lake Superior have at least one unsafe day per year due to fecal contamination. So it's a problem. And then there's the question of, well, what's the source of this fecal bacteria? And there's arguments on whether it's beachgoers, dirty diapers being deposited, whether it's animals or wastewater. There's all sorts of questions around this. So it's a very interesting problem, this beach monitoring and what the sources are. And it's an important health issue. So next slide, Stephanie. So
yeah, so this is our one of our first polling questions, just to keep everyone awake and engaged in the webinar. And so, as I mentioned at the beginning, you just navigate to the pollev.com /SOTS549 at the top. If you've already been there, it should reset, kind of refresh, or you can hit your refresh button and just type your answer.
That's a great question. I like to see these answers. This was when we started this, Sea Grant approached me in June, and there were a lot of questions. And this was one of the big questions. If it gets in the water, how long does it survive? And it depends on the lake temperature. There have been studies of other coronaviruses that ask this question, where people, spike coronavirus into lake water, and at room temperature 25 degrees Celsius, most other coronaviruses don't survive very long, maybe 24 hours. But what they found was, as temperature goes down, of course, you preserve the virus, and intact virus could then survive for up to a week or so of testing. But still, it's not been tested with SARS-CoV-2, so we don't really know. I think we've covered the whole range of.
I think we've got all kinds of answers here, right? Exactly. Thanks, everyone. This is great.
Yeah, and what we think we understand now about the survival of the virus in the waters, as Dr. Prather said, it doesn't want to be in a water environment. It really wants to be inside of a cell. And
I forgot we had, okay. Sorry, I'm gonna make everybody seasick. I forgot we had two questions back to back, but please continue, and people can think about their answers to this one as well.
I don't see my next slide, but that's all right. Anyway, I was saying that now I think that thinking about the mode that we were thinking about SARS-CoV-2 entering the water, which was potentially from beachgoers and fecal contamination. It's thought now that the virus doesn't, even though it infects epithelial cells of the intestine, it doesn't survive intact through fecal matter. So we're going to be thinking about fragments of the RNA genome that survive in very small fragments. So, we know that the virus is shed in feces of infected people for up to 2 weeks after they're done with the respiratory symptoms. So you could have beachgoers coming who had previously been infected, and then through fecal matter washing off them while they're in the water, leaving some SARS-CoV-2 in the water. So we were thinking that this would be primarily a way to say something about the population that was visiting the beaches, and then give feedback to the tourism community and beachgoers about their relative risk at the beach. And as I have here, we now know, months and months later, that the fecal oral infection route is very, very rare. So this is another lovely picture of the beaches at Duluth, Minnesota. So next slide, Stephanie. So I monitored 8 beaches in Duluth, Minnesota. So you can see where I've marked Minnesota Point here. I monitored 5 beaches along this 7-mile-long sand spit. It's very, very popular. Gets large crowds of beachgoers during the warm months. And then I had 3 sites in the city of Duluth, and these tend to be more neighborhood kind of local beaches. And then as you leave Duluth, you have a lot of tourists going to that last star marked at the top of the map there. The Minnesota Point goes across this bay. There's a lot of shipping activity in there, and there's the St. Louis River estuary off to the left. And that's where sewage, treated sewer water will enter from both the city of Duluth, which is about 100,000 population, and the city of Superior, which is somewhere around 30,000 population. So we started on July 4th with large crowds, and we monitored every weekend, 3 days consecutively over the weekend, Friday, Saturday, Sunday through November. And we'd collect 100 milliliter samples. We'd filter out the bacteria. We'd pellet the virus with polyethylene glycol precipitation. And then we would purify small RNA because we suspected that the virus was being fragmented. And what we found is that we get a lot better detection if we go after small RNAs. And then we just use the standard CDC SARS-CoV-2 PCR test that we use for our wastewater testing, and that's used for people. And we also importantly included the pepper virus, pepper mild model virus. And this is a virus that infects peppers, but it's in most of the processed food that we eat, and it's the most common virus in human feces. So it gave us a way to say that it's also an RNA virus. So it gave us a way to say that we were actually purifying virus. If we didn't detect SARS, we would expect to maybe detect the pepper virus as an indicator of human activity or presence of humans. So next slide, Stephanie. So this I just always want to be clear that we are looking for fragments of the SARS-CoV-2 genome. We are not detecting intact virus or live virus. And so I just put this slide in here to say we use a quantitative PCR test that looks for one small part of the genome, and that's what we're detecting. Next slide.
This is our polling question for the pepper virus.
Yeah, the pepper virus was something that we thought was important just to prove that we could extract virus.
Well, it looks like everyone agrees.
That's good, thank you.
For it, that's great. Yeah. Oh, we got a maybe.
Yeah, that's. So I don't want to interrupt voting. Okay, so this is the results of the measurements that I found. So on the y-axis is the log of SARS-CoV-2 genome fragments per liter that I was detecting. And then the sampling dates are on the bottom. I have a little bit of British background, so it's day, month, year. And what you see is that up until September of 2020, I detected nothing for SARS-CoV-2. There was not a trace of a signal. We let these PCR reactions go out to 50 cycles to try to pick up anything that we can, and nothing ever showed up. However, we did pick up pepper virus, so we were confident that we were extracting virus and that if SARS-CoV-2 was there, we would see it. Then you see this dotted red line, and this was a big shocker to us. This data is from those 3 beaches in the city of Duluth. And all of a sudden, at the beginning of December, we saw this huge signal from SARS-CoV-2. And it coincided with an uptick in the pepper virus as well. And so we saw this huge uptick in SARS-CoV-2, which then gradually tailed off until the end of our sampling in November. We were not detecting any SARS-CoV-2 anymore. The pepper virus was still pretty high. And then on the next slide, it shows the Minnesota Point beaches, the big long sand spit. So there as well, we saw virtually no SARS-CoV-2 all through the beach going season when there were people in the water. And then at the end of the season, you have to understand in Duluth in September, the air temperature is pretty nice, but it's not nice enough to be in the water. And we saw a little bit of increase in pepper virus, and then we saw some minor increase in SARS-CoV-2 detection. Now the blue line is for one of our probes, which is a bit more sensitive, and with our, but it's not quite as reliable. So if I don't see both of those probes coming up, I'm a little cautious about calling it a detection. So we had a hint, but it's hard to say if it was a serious hint or not. So next slide, Stephanie. So those were our findings. So what we conclude, we saw nothing during beach season. So we don't think that beachgoers are a significant source of SARS-CoV-2 in recreational waters. So we think that it's rather safe. Your major risk on the beach is going to be infected people on the beach, not SARS-CoV-2 in the water. We're still not quite understanding what the source of the spike that we saw. As I said, the most likely source would probably be sewer leaks or sewage, and the sewer plants in the area release a treated sewage into the bay, and then those currents travel up the Wisconsin shore, not up the Minnesota shore. So we have some interest in doing some source tracking. There are some rivers that run in to the lake there in Duluth, and they could be sources, either hospital leakage or septic tank leakage. And then I think that going forward, because the SARS-CoV-2 pandemic will hopefully be over soon, what we are really interested in doing is exploring use of the enteric virus as more of a tracer of human activity, and seeing how things like sewer leaks or even effluent from the wastewater treatment plants flows and is diluted into Lake Superior. So we're using the pepper virus and then this other human enteric virus, crassphage, and caffeine, which is pretty commonly used. And so that's our results. I guess next slide, Stephanie. And so I just want to say that during COVID pandemic, it's not easy to assemble a team, especially when you work mostly with students who are now remote. I had some luck in I didn't find biologists or medical students. I found outdoor education students who were very dedicated and very concerned with the environment and helped me out and learned a lot of the lab techniques as well as going out into the field. And then I want to thank Minnesota Sea Grant for their support for the project. And so any questions, I'll be happy to address.
Great. Thank you so much, Karen. I think we might have time for one or maybe two short questions.
Okay, the first question that came in from Katie was that wanting to know, were there any notable rain events during the later half of the sampling period, i.e. October to November compared to July and September?
Yeah, that was what was so strange. It was a particularly dry year in this part of Minnesota. So there were no major rain events that coincided with that spike. It was a very strange spike.
Okay. And Sam was actually asking, I think it's probably the same thing, so that you didn't see a spike in the holiday gatherings at that time either.
No, that was at the very end of August. So compared to 4th of July at the beach is pretty huge. But this would have been around Labor Day. And you just don't see the kind of crowds at the beach that you do around 4th of July like that. It's a little too chilly in Duluth at that time for beachgoers.
Okay. Sam also had a great clarifying question that others may be wondering too. You mentioned the COV2 into. Could you tell us a little bit more of exactly what that is?
Yeah, sure. So in that slide where I was showing the genome, the CDC assay that we use, it looks for 2 different locations in the N gene. So the N gene encodes the nucleocapsid, and that's a protein that wraps around the RNA genome. So we look for 2 different sites in that gene. The kind of standard wisdom is that you need to have both of those detected in order to call a positive test. So that's what's done with human clinical tests for the most part. You're called positive if you get a positive N1 locus and N2 locus in your test.
Okay, great, thank you. Stephanie, do we have time for one more, or do we need to move on?
I think we have time for one more.
Okay. Michael was asking if you think the research is robust enough to extrapolate this to other areas, i.e. warmer climates, or maybe even different seasons.
I'm really confident in the methods. We spent quite a long time with our wastewater project to develop those methods. We think they're pretty sensitive. I was surprised that they were transferable to lake water. So if they're transferable to Lake Superior water, I think they're pretty robust.
Okay, great. Stephanie, we do have one more. What do you want to do?
Again, well, we can do it. I think we can make the time. Yeah.
Okay. Tony was asking, he's identified that he's actually down where the Rio Grande drains into the Gulf of Mexico, and sewage treatment is doubtful there on the Mexican side. So he's wanting to ask if when fishing at the river mouth, should he be concerned at that point?
So what we understand about the virus when passing through the human body through your intestine is that it's unlikely to survive your intestine. It's going to come out in fecal matter in fragments. As Dr. Prather had said before, no one has really tested whether there's infectious virus coming in sewer water. The thought is it's unlikely, but it's not been tested yet. So.
Okay. Thank you very much. And Stephanie, we can definitely move on now.
Good. Thank you. Oh, we must have missed a slide.
No, that's right. Thank you. Thanks for listening. Thanks.
Great. Thank you. So our third speaker is, I'm probably going to get your last name wrong, Heather Prendizan. From Penn State. So I'll let her fix her last name and turn things over to you.
No worries at all. Everyone has the same struggle, so it's totally fine. So my name is Heather Prendizan. I'm an associate professor at Penn State in the Department of Agricultural and Biological Engineering. And I just wanted to say thank you to the organizers of this webinar and of course to Sea Grant for funding this work. We're very excited to be part of this project and to be sharing what we're doing with you all today. So you can move to the next slide, please. So I think we have our first poll question.
Polling, yes.
Yay, right off the back. So this is just thinking about what you do every day and how the decisions that you make throughout the course of your day might affect what winds up in your wastewater.
I was going to say everybody was overachievers at the 100%.
And it's totally
yes.
fine if you say no. I would hope that nobody spends too much time, except for those of us actively researching wastewater, thinking so much about it.
Good. Great. Well, it looks like the numbers have stopped moving. So we've probably gotten most people's
Okay.
answers. But
Great. Well, I guess you can move on to the next slide, but it's great to hear that the majority has. And for those of you who haven't, I hope you're not too grossed out by the following presentation. So just to do a little bit of an overview of what's in wastewater, if you think about a wastewater treatment facility and sort of the area that it services, you can think about your domestic sources, so your household or your neighborhood, and all the things that you do in your household. So between flushing toilets, bathing, using your kitchen sink, laundry. So these are all sources of domestic wastewater. And so you can think about how, you know, for those of you who haven't thought before, perhaps about what products you're using for household cleaning, your personal care routines. Of course, those are going to wind up in your wastewater. And then industrial and business sources of wastewater in the sewer area, as well as hospitals, if you happen to have a hospital that's discharging to a wastewater treatment plant. So those, if you think about from a wastewater treatment plant's perspective, if you're the operator, those are all the sort of factors that are contributing to what shows up at the wastewater treatment plant on the influence side. So next slide, please. So it's lots of stuff that winds up in the wastewater. So pathogens, viruses, bacteria, parasites, those are all, of course, human health concerns. Nutrients, so nitrogen and phosphorus are also in the waste stream. And if you think about, for those of you who maybe use composting for your gardens, biosolids, which get generated through wastewater treatment plants. So the solids waste can often be recycled and repurposed as a fertilizer source. So utilizing the nutrients that remain in the wastewater, oftentimes you see effluent irrigation, especially in more arid climates, as a source of beneficial reuse water. So leveraging the fact that there's nutrients in that wastewater and then using it for agricultural sources of nutrients. So those are all good benefits. And then of course, chemicals, so domestic, industrial, and hospital sources of chemicals. So personal care products, pharmaceuticals, prescription and over-the-counter medications. So a lot of those things wind up in our wastewater treatment plants as well. Next slide, please. And so we can think about wastewater, I guess, in sort of the ick factor, but there's also lots of ways it could be useful. So can you think of, we've touched on the nutrient perspective. So what are some ways, other ways maybe that wastewater could be useful? Yep, definitely fertilizer, great. This is fun. I've not used one of these poll everywheres before, so it's fun to see this. Yeah, energy sources, yep. Just letting it evolve. It's actually kind of hard to track the same word as the slide evolves.
Yeah. I guess as it gets bigger, there's more than one answer.
Yeah, but lots of things around beneficial reuse I'm seeing. Recharging of groundwater aquifers, sure, if you're doing irrigation. It's awesome.
Such engagement.
Yeah.
It's so nice. Great, everyone. I'll be looking. Will be nice to do this graphic at the end.
Absolutely.
Afterward.
Oh, that's so fun. Great.
Great.
All right. Whenever you want to go to the next one, I think we're ready. So one of the things that we were hoping to use our project for was wastewater surveillance, and to use what comes into the wastewater treatment plant as sort of a metric of the health of the community. So you've heard a couple of the other speakers talk about using this to track specifically the SARS-CoV-2 virus so that we can sort of get a community scale assessment of the physical health of the community. So trying to understand, especially when testing resources are low, what the potential bigger scale of the problem might be in your community. And if you're sampling frequently, then you might be able to get ahead of the curve and maybe make some useful decision making at a community scale about, you know, how much to reopen and things like that, were sort of the vision of how this was thought to be useful from sort of a decision making perspective. But there's also, and this is, I guess, where my research area of interest really drove this project, was on the pharmaceutical side. So my research group focuses a lot on the presence of emerging contaminants in wastewater and what the ecological impacts of those might be if they persist through the treatment plant. And so we started to think about, okay, well, there's going to be a lot more people potentially taking over-the-counter medications or prescription strength medications, or these new sort of experimental drugs that are being used in hospitals that could, you know, wind up in the wastewater treatment plants and then persist through to the effluent. And, you know, there's concerns on the ecological impact side, but if you can measure those on the influence side, then you might be able to look back at the physical and mental health of the community over time. So we wanted to look at things like antidepressants and anti-insomnia drugs and things like that, to kind of get an assessment of maybe how stressed out people are, and how, you know, all the pressures that we've all been feeling as our lives have been so greatly disturbed and changed over the significant part of the past year. That's obviously taking a toll on everyone. So this is a really good way of seeing wastewater as a source of information that can be used to really understand what's happening at the community level, not from only a physical perspective, but from a mental health perspective as well. So next slide, please. So we have already been started to work on this on campus to look at the Penn State wastewater treatment plant, as well as the broader communities treatment plant called the University Area Joint Authority. And we had been funded through seed grant opportunities through the College of Ag Sciences at Penn State. And I got a call from Pennsylvania Seed Grant folks asking if we would be interested in scaling that work up to potentially look at other treatment plants across the state. So really what we wanted to do was expand what we are already doing and provide the opportunity for more wastewater treatment plants to work with us, and to give them data back in sort of a rapid way so that they could find it to be useful, hopefully for decision-making purposes. So our goals were to provide data that could give wastewater treatment plant operators and municipalities or organizations information about population assessment for SARS-CoV-2 presence, and then potential changes in the mental health and physical health of the community over time by looking at different pharmaceuticals and tracking those over time as well. So this is some information about our project team. Some of us have worked together for a long time. Others of us have never worked together except over Zoom. So that's been really interesting to build a whole new team of collaborators sort of in these remote working environments, and to just make it all happen with challenges with getting students in the lab and having you know, less people per square foot than we can normally have in the lab. So it's just been crazy to try to orchestrate all of that and to pull it all off. But we've had wonderful collaborators at the USDA ARS who have also helped, especially on the analytical chemistry side. They've been able to help with some of the pharmaceutical analysis that we've needed help with. And then the collaborators that we're working with right now are the Department of Corrections and Derry Municipal Township are our two partners. We had originally proposed 3, so we are still looking for a third collaborator to work with us through. We have funding through August, so we're hoping to add a third just to round out what we had originally proposed. And then sort of our broader partners are the Susquehanna River Basin Commission. We have a partner at Harrisburg University as well. So next slide, please. So if you're curious what this process all looks like, and so you've seen a little bit of this perhaps in the previous speaker slides, but basically there's someone at the treatment plants that's pulling regulatory samples anyway on their influence. So they just pull an extra sample for us and it gets shipped to us overnight. And then it goes right to a biosafety level 2 lab because it's got pathogens in it. Even if SARS-CoV-2 isn't infectious, there's clearly other things in the wastewater that we don't want our students to be directly exposed to if it were to come directly into my lab, which is not a biosafety level 2 lab. So it gets pasteurized first to inactivate anything that may otherwise be infectious in the sample. And then it gets extracted from by the virology team for basically the genes of the virus. So the same analysis procedure that was described previously. And then it comes to my lab where my students further extract the sample for pharmaceuticals. So you can kind of see my team getting very excited to do an essential project because that was one of the justifications for reopening our lab. So they've been really happy to have this project in our portfolio so that they can actually physically be there. That all happened far sooner than it would have otherwise under university procedures because this was deemed an essential project. So they were very happy to be back in the lab and doing some very relevant and exciting work. And then what happens with that data is it gets compiled, both the virus data and the pharmaceutical data. Basically, I get an email with the data and then take it and compile it and send it out to the plant operations engineers. And hopefully they're using it to do some sort of decision making at the community scale, though I wouldn't know what exactly they're doing with it. So it's just sort of been a one-way exchange of information. They do send me the flow rates so we can get information about the flow conditions at the treatment plant. But basically, it's just us reporting to them what we're seeing in their samples. Next slide, please. If you're interested in what pharmaceuticals we're looking at, here's a list. They're numerous, so I won't belabor the whole list, but you can kind of see them categorized. But over-the-counter medications related to minor symptoms that people who have been infected may be feeling or experiencing, a suite of antibiotics as well. Sometimes if they're effective, then obviously the person wouldn't necessarily be infected with the virus because it wouldn't expect an antibiotic to be effective at treating someone who's infected with the virus, right? So we are looking at that as well. There has been increases and antibiotic usage through this process, especially as people have secondary infections as a result of being sick. And then we also looked at hydroxychloroquine and remdesivir. We haven't seen hydroxychloroquine in any of the samples that we analyzed, so that's just been interesting. We have seen remdesivir, though, especially when some hospitalizations have increased in our communities where we're collecting samples from. And then we started creating procedures for some of these other more mental health indicators, but they weren't things that my lab group had been analyzing for previously. So it's taking us a little bit of time to get that up and running. But I believe we're starting to get there in terms of getting the methods that we need up and running on our analytical equipment to be able to start reporting that data back soon. And this is just a snapshot of some of the data that we've sent back to the treatment plant operators so far. So acetaminophen and naproxen are pain relieving medications. So we're reporting concentrations in nanograms per liter, and you can see weeks across the x-axis and concentrations on the y-axis. And then remdesivir, of course, is one of the experimental drugs that's being used in hospitals for treating pretty severe patients. Next slide. And again, some of the results so far, you can kind of click through this if you'd like. The top table is we have a shorter period of record for this particular partner. And the graph on the bottom is from a different treatment plant. So we've got two partners. I just have taken out the information for which one is which. But this is basically the information that we've given back to them so far. We have uncorrected and corrected concentrations that we're sharing. One of the things we've noticed is that the recovery is very variable for the wastewater samples that we've been collecting. So we monitor that, we spike the samples and can get a recovery factor to use to convert basically an uncorrected concentration to a corrected concentration. So we've been doing that and hopefully that's useful for others. If you're not doing that and you'd like to talk to us about that, I'd be happy to share what we're doing. and maybe talk about issues that we're having with recovery factors being sort of all over the place, which has been just, yeah, why we're doing it for every sample so that we have it documented and can account for it. And I mentioned that we get the daily flow data as well from our treatment plant collaborators. So basically they give us the flow rate, we give them the viral copies per day from the sample that they've collected. They're usually doing 24-hour composite samples. So they take a small subsample over the course of the entire day so that the sample represents a full day of effluent. And then we can give them basically the load of the virus. So how many copies of the virus per day are passing through their treatment plant. So that's the information that gets sent back. And these on the right side of your screen are some of the things that we're hoping are some of the outcomes. Some of them we have control over, others we don't, but we're hoping that this data gets used to correlate virus prevalence in the community to serve as an early indicator of rising cases or falling cases in some cases, target testing resources. And I think what I'm hearing at least from our treatment plant partners is that they really like having their finger on the pulse of this problem, that this just gives them information that maybe they can't act on, maybe they can act on, but either way, they seem to feel better knowing what's there. So if nothing else, at least they feel like they've got their finger on the pulse. All right, next slide, please. And I just wanted to take another moment, of course, yeah, as said by previous speakers, researchers, research doesn't happen without our students, our amazing students that make it all happen. Otherwise, it's just ideas on a Word document on our computer somewhere. So they actually bring all the research ideas to life and make it all happen. And of course, funding and collaborators who are willing to pull the samples and send them to us. So just wanted to thank everyone who's been a part of this team so far. And if you're interested about the things that we do that are not related to COVID and wastewater, you're welcome to check out my website. We have a cool little footprint calculator that you can fill out if you are interested in knowing what your household's footprint is from an emerging contaminants perspective. So if you would like to try that out or have any questions about that, you can also find that on our research website. So with that, I'll say thank you, and I'd love to talk more if there's time.
Thank you. Thank you, Karen. I think we're only a few minutes behind. I think we have time for one question.
Okay, well, first of all, Mark with Georgia Sea Grant had offered that there's a website that actually has information on what they're pulling from COVID for their local sewage treatment plants. So Mark, if it's okay to share that with everyone, could you put that in the chat for us, please? And then Sam had also indicated that the pharmaceuticals being detected might also give us some insights into the accessibility for the newer treatments and help us inform social equity needs. Would you agree with that, Heather?
I'm sorry, I missed one of the words that you said. Could you just repeat the last part?
That the insights would maybe help with accessibility for newer treatments and help to inform social equity needs.
Okay. Yeah, I think that would be great, especially if you have information from lots of different kinds of wastewater treatment plants that service areas with diverse communities is in their service area. So I'd love to see more of this data get shared. And yeah, think about environmental justice issues as well. So I think that's really important.
Okay, great. Well, thank you very much. And Stephanie, I think we can move on to the last presentation for right now.
Thank you.
Thank you. This is so much information. I think it's just been great so far. So our last speaker for today is Chris Stepanek from Lake Champlain Sea Grant. And this was kind of highlighting, not just the research, but kind of what are the questions out in the field when things like this come up. So Chris, I'll turn it over to you.
Thanks very much. So just to center us, if you want to go to the next slide, please. Thanks. To center us for the conversation, the realm that I'm speaking from is talking about volunteer water monitoring programs and where and how they've been impacted and what they've reacted and how they've reacted to the COVID situation. So what we know right now is there's about 1,700 programs across the United States that are supporting volunteers to engage in water or water related monitoring, whether that's habitat or wildlife, aquatic wildlife. We know that there's about 83 of those programs are supported by Sea Grant across the nation. And there's about 100,000 volunteers, 16,000 or so of them are working within Sea Grant supported programs. And these programs are doing things and having outcomes for both the people who are participating and the communities in which they operate. So that's everything from increasing scientific knowledge of the people as they collect data, and also contributing to our understanding of the natural world. It's building people's social networks and their opportunities professionally. These programs are giving the volunteers an opportunity to engage in decision making and have a voice in decision making, and they're affecting policies and natural resource management decisions. So when COVID hit, next slide, please. I've been working in the field for a couple of decades now, and have worked on a national scale for trying to integrate and help volunteer water monitoring programs across the country integrate with one another. And so I started getting emails when COVID hit and everything was shut down last spring. You know, what are other programs doing? What should we be doing? From local coordinators of programs or statewide coordinators. They recognized that there was recommendations even from the CDC and others that said that being outside is a safe place to be, and these programs are outside. They realized that people needed some kind of normal in their lives, and they wanted to try to maintain that if they could with their programs. They recognized that sometimes people were actually looking for something to do because they had time off, or because they had their families with them all the times and trying to do something with their family that could be valuable to the environment, valuable to the family, and an opportunity to be in a safe place outdoors. And they also realized that oftentimes the volunteer water monitoring program methods could be done safely within a household or a family group, and safely distanced from other people. There was also the recognition that some of these programs are 20, 30 years old, and they had long-term data sets that if they could safely maintain those and continue to collect data, they would like to do that. And so next slide, please. I was contacted by Jill Carr with the Massachusetts Bay Estuary Partnership, and as one of those folks. And I said, you know, I've been asked by enough people, maybe we should have a webinar and pull people together to try to brainstorm what we should be doing that would make sense to be able to continue having these volunteer monitoring programs throughout the 2020 season. So we gathered folks in late April. We had a lot of interest. We had almost 300 people register for the webinar. We had over 200 participate. This webinar generated about 90 recommendations that were broken down into categories. What do we need as coordinators of volunteers in water monitoring programs to be doing to continue to engage these people? What should our field crews or our volunteers be doing before they leave home, when they're in the field, and when they're in the lab? And so if you go to the next slide, I've broken it down into three categories of recommendations that were arrived at through conversation during that webinar and pre and post webinar. And I say base, space, and place. And so what do I mean by those? In terms of base, one of the things that came up is relying upon the seasoned volunteers rather than bringing in new volunteers who it would be difficult to train them and to engage them safely, and utilizing the services of the volunteers who are already participating in the programs. Thinking about modifying communications approaches with the volunteers, for instance, updating program guidance to include COVID-related modifications, facilitating online meetups. People realized that their volunteers were not necessarily having the opportunity socially to connect with people, and so connecting in a new way about the program was something. Having social media sharing among the volunteers about what they were seeing if they were going to continue monitoring. Offering online question and answer sessions, developing communications plans with those volunteers if they were not able to monitor due to illness or the inability or concerns over participating. By space, some of the things that the suggestions that came up were to change the field teams. So again, thinking about households and who can participate together, thinking about the timing or logistics. So at, if you use a water site, if you say a stream site, maybe in the same day, the different household teams would participate one after the other in time, but not at the same time as one another, to try to space out who's participating when. They also were coming up with tips for one another about modifying field sampling methods. There's one program I know that went completely virtual. They were looking at fish spawning, and they were able to install cameras, so the volunteers were able to just watch the camera and see the fish or what they needed to see with the way that they had set up the cameras to be able to watch. They also modified lab setup to support physical distancing, installing partitions, having one-way ingress and egress within the labs. Some of the programs removed some types of sampling, or they assigned a family or a household group to specific equipment, so they didn't have to share equipment. This was before we understood the mode of passing of the virus. And then place, thinking about conducting training online. And that's one of the biggest things that programs did, was to shift their trainings online, utilize videos, utilize Zoom, like we're all doing these days. And then doing different ways of getting samples if they needed to get to a lab, driving around a geographic area and having volunteers drop off to a trunk, so a contactless handoff. If you are interested in seeing the tips and the tricks that the group came up with, if you use this website that's shown here, all of them are available. And there are a number of resources that people shared from what they put into their guidance for their volunteers to videos that they made that are available in a Google shared document there. Next slide, please. So one of the things that I did after that webinar was to just send out a survey to the groups to try to understand what kinds of impacts that COVID had on the programs to date. And the survey was sent out in June. For many of the programs, that's early in the field season. And I'll focus on this slide, just on the blue dots, the chart, the bars, whatever they're called. So I asked a number of questions to try to summarize the kinds of advice for the base space and place advice that I was just talking about. And so we know over 70% of programs had already implemented by June guidance about COVID modifications for their programs. About 60% of the programs had postponed their field season. More than half of them had done something around changing those field teams or the timing about when they would go out and monitor, and just under half had changed their communications approaches with volunteers. So again, the examples were offering social networking opportunities through Zoom, or not having in-person trainings. And then next slide. On the other end of the scale, one of the things that we weren't sure what would happen at the time of the April webinar was if the programs would cancel their field seasons. In the end, less than 1 in 5 programs actually canceled their field seasons for the year, for some of those reasons that I said earlier about recognizing that being outdoors is a safe place, wanting people to be able to have the opportunity to have some normalcy in their lives, and being able to continue those long-term data sets. The next most unlikely thing that that groups had done was about a third of programs opted to only rely upon their seasoned volunteers rather than bringing in new volunteers. Next slide, please. I also asked the programs, what else would be useful? One of the things we realized during that April call was that there was a lot of questions people had, and they were very interested in getting back together. So we ended up having a series of 5 webinars for the program coordinators. We had almost 900 people participate, and we relied upon the Sea Grant Network heavily. So 2 of those webinars were offered by Lisa Tassi from Maryland Sea Grant, where folks were wanting to know how to communicate better using digital tools that were available. And then Sam Chan and his team did extensive research to try to understand what was known to date. And this was from September up till September of possible presence of the virus in surface waters. And there was a lot of interest around that. Next slide, please. I also asked programs what kinds of negative impacts they had. and they reported a variety of different kinds of impacts. Many of them are reporting, over 70% reporting, that they had fewer data observations made than in a usual year. More than 60% had volunteers numbers drop. And again, partially based on the fact they couldn't train, partially based on the fact that they were just choosing to utilize the services of the seasoned volunteers. Just under half of the programs had some kind of economic loss. And when I dug into that a little bit more, the good news is that most of the economic loss was for less than a quarter of the overall budgets. And some of the programs were able to shift budgets around to support staff to do other things besides what they might have been doing in a normal season. But there was about a third of programs that did lose staff or staff time as a result of the COVID experience. And next slide, please. And then the last thing that I asked was to try to understand, was there anything positive that had resulted from the experience of COVID and what it had done to the seasons to date? Again, this was in June. And so there was over 100 people who answered the survey, and I think about 40 of them came up with something positive to share. The size of the words here on this Wordle mean that was said more often. So one of the biggest things was that they were able to develop distance learning tools that they would be able to utilize over the long term. Some of the programs had increased participation, again, because of people's interest in trying to do something meaningful with their families or households over time. A lot of the programs were able to take a breather and think about what they were doing and using the opportunity to assess what they were doing and to respond. And the program coordinators who answered the survey felt like they had increased their resilience to be able to take what comes as it goes, partially related to the developing of the distance learning tools, and partially because they were able to weather the storm through with their economics and staff loss and volunteer loss and whatnot. And that is the summary that I thought I would share with you for today from the volunteers' perspective.
Thanks, Chris. So I thought we had a couple of polling questions from Chris, just as a kind of transition into some of the discussion we wanted to have more focused on the Sea Grant kind of network. But Karen, we may be able to, like, if there's particular questions, we can probably do that at the same time we're doing polling.
Sure, I can try to negotiate that a bit. Tina was actually asking, Chris, have you published your research at all?
I am submitting it next week. It is 99% of the way there, but not yet.
Okay, congratulations.
After next week.
Hey, you've got 99% of the way done. So there's a variety of really great questions. Did you want to just stick with Chris right now, Stephanie, or go back to some of the other questions?
I think we can probably go back. I mean, I think that'll be okay to go back to some of the other ones. So I'm just going to give this one more second. And then the next polling question relates to, again, to Chris's presentation, but I think other presenters can field questions as we go on.
It's exciting to see not looking to cancel or postpone yet. It's good.
One question that I think is probably pretty easily answered for Dr. Prather. Has anyone extended the research that you've been doing in other areas, i.e. in the Gulf of Mexico or maybe even in the Great Lakes, any freshwater systems as well? Kim, you're muted.
Yep, got it. Yep, sorry. Not that I'm aware of. And like I always just like focus on San Diego, like sewage is going in the water, and that goes in everywhere, right? And as far as I know, I have not seen anybody looking for the, you know, I've looked for, there are people who are looking at, you know, certain toxic blooms getting into the air in lakes, right? But not SARS-CoV-2 that I'm aware of.
Thank you.
Mm-hmm.
Stephanie, do we want to field the question on the screen or keep moving forward?
I I think we can keep moving forward only because I know we're very close to our time. So this is great. And we will be sharing the polling results with participants and the panelists afterwards as well. So we really do appreciate all of your participation.
I'll go ahead and follow up with one more question for Dr. Prather. The question was if the virus has actually been found in marine organisms, and has there been any research that might indicate that they've been sensitive to the virus?
No, that is a great question. And no, not that I'm aware of. That is something we should all be looking for, right? It is, I will tell you that in the estuary, in the river, it is there. You know, it's flooding in, you know, to those environments. And so, you know, ocean organisms are experiencing this virus. And I don't know of anybody who's thinking of it right now, and they should be.
Well, thank you very much. And everyone, you've had phenomenal questions throughout the entire presentations, and we really appreciate your involvement. Stephanie, do you want me to hand it back over to you?
Yeah, that sounds great. So thank you so much for sticking with us through the whole webinar. And so we just wanted to have a few last minute questions to get the Sea Grant Water Resources Network kind of thinking about what these presentations might have sparked for you or your program. And so we only have a few minutes for these, so you may have to think fast. So this is our first webinar. We're kind of learning about the timing, but the first one was just, you know, did the presentation spark any ideas for you that your programs might pursue? I think we may have stumped them, Karen. Yeah.
Find that hard to believe.
Right.
Yeah. And please feel free to put stuff in the poll everywhere. And if you have things that you might want to engage with the panelists about, feel free to put those in the Q&A or the chat as well.
Great. Really good ideas.
These are really good.
Okay, well, they seem to have slowed down a bit, so I'm going to go to the next one. And so we wanted just to think about, like, if, you know, we heard a lot about some of the individual research projects that might have been happening on a discrete geographic level. And so we wanted just to think about if there's anything that could be, you know, scaled up a little bit, regional or national.
Stephanie, I think one thing for sure, as we thought might be the case, we're going to have more questions than answers when we're done here.
Yes, yes. And then, so for those who are involved in the Water Resources Vision team, we are going to be collecting all of these ideas and in thoughts and things that spark from the webinars, taking them back to the team and really having more conversations about what we can do on next steps. Another webinar. We have so many ideas for webinars. Yeah. Good. And so this is our final question. Hang in there. Everybody's been so great with great participation. And so just, you know, thinking about what is special and unique about Sea Grant and what can we kind of focus on. Yay, well, positive feedback. Like that.
You know, Stephanie, the outreach help answer just makes me think, you know, as the panelists here, do any of you have ideas for particular types of outreach that might be helpful, you know, in the communities that you've been working at?
Oh, I think Richard's results are pretty interesting, and it would be really interesting to see if it holds true in other places, and to tie them into with Kim's in terms of what's the aerosol connection, but the lack of in the water at the beaches is pretty useful. I think that people should know.
Yeah, Minnesota Sea Grant has a website. It's specifically for the Park Point people, and we post everything like riptides and bacteria concentrations and things like that.
Great. Well, thanks everyone so much. These presentations were great, and I think this was just a really great way to kick off our webinar series. So I want everyone to know, to stay tuned, that we are making plans for the next couple of webinars. We are hoping to do at least 3 this year. If we get ambitious, we might do 4. We're not sure. The next one that we're working on would be focused on emerging contaminants in kind of the April-May time period, and then later this year, stormwater, green infrastructure focus. And if you want to know more, please check out our vision plan. It's on the Sea Grant website listed here, but you can also just Google like Inside Sea Grant for that, and it just gives more details about what we think some of the research needs, outreach needs are as well. And again, just a big thank you to everyone. If you want additional information, please reach out to me. I really appreciate everyone's attention today. Also look out for a survey link that'll be coming. So Zoom will send everybody an email thanking them for their participation, and there will be a survey link in there. And we would really love to get your feedback on this first webinar series. So everyone have a great evening, and I really appreciate everyone's participation.